The broadcast is now starting. All attendees are in listen-only mode.
Hello everyone, and welcome to Lab Manager's Tech Trends webinar series. I'm Tanuja Kopal, contributing editor for Lab Manager, and I will be moderating today's discussion on the use of mass spectrometry for food safety analysis. We are joined today by two technical experts from Agilent Technologies and Thermo Scientific, who will be talking about applications of mass spectrometry as it pertains to the food industry. And they will also point out the types of instruments that will best serve certain analytes in certain applications. We try to keep things very interactive and informal, and we would like you to make use of this live opportunity to pose questions and have them answered real time by our experts. And you can send along your comments or questions anytime during the webinar by typing it into the question box that appears on the right-hand side of your screen. We will address your questions right after our speakers have gone over their brief presentations. Today's webinar will also be available for viewing on Lab Manager's website, which is www.labmanager.com for at least 6 months. The website also hosts a number of resources such as application notes, white papers, and links to all our past webinars, which you may find very useful. In our closing slide, we will also share with you the email addresses of our speakers, so you can reach out to them for a copy of their slides or for further guidance on this topic. You will not be able to download slides off of our website. So with that, let me turn it over to our first speaker, Paul Zavitsanos, who oversees the worldwide food safety market development team at Agilent Technologies. In the past 25 years, Paul has worked in various positions within the chemical and analytical instrument industries. Paul has extensive hands-on and training expertise in HPLC, GC, GCMS, LCMS, and LCMSMS with both trap and linear quadrupole instruments. So welcome, Paul, and look forward to hearing what you have to say about mass spec for food safety.
Thank you very much. And we'll head off to the first slide. I want to thank you all for coming to this presentation. And I need control of the slide set here. It doesn't seem to be allowing me to move my slides. Hello?
All right. No, we'll take care of that. Let's well, try to end it and open it again and see if that works. Any luck, Paul?
Nope, nothing at all.
Well, just right-click on the top to end the slideshow, and then maybe if you reopen it again, it should do it.
Right-click at the top.
Yeah, where you have the X on the box. All the way in your all the way in the right-hand corner, where you have the X to close the slideshow.
All right,
yep.
I've got that now, so let's open it again.
Yep, that should do it.
Let's see what happens. Look at that. Okay.
All right.
Thank you very much. And this is the way my day has gone, because at first thing in the morning, the hotel that I was in had no Wi-Fi. So this is the morning for technical problems. So I want to thank you for your confidence and for your patience. So my presentation is about the food safety efforts of Agilent Technologies, and how we see the food safety market, and how we also take a look at what kinds of mass spectrometers could solve what types of problems in the food safety arena. So first thing we have to ask, and hopefully for those of you on the line, the ones that are in the food safety business, this is old hat. You understand this. Food safety is becoming increasingly important because of the increase in the globalization of food and the change in the food laws. And a lot of this is driven also by pathogens and the fact that outbreaks are happening in the United States very frequently, and in the rest of the world very frequently with severe consequences, and the chemical impacts as well of pesticides, mycotoxins, and other allergens, and marine toxins are rather severe. So all these issues are combining to form an environment where increased pressure is put on the technical measurement and the quantitative measurement of some of these agents. And we've been doing this for many years, of course, as have most instrument companies. We've been doing pesticides and veterinary drugs and trace metals for many years. We've had extensive experience with pesticides over 40 years, with electron capture detector, etc. The difference is that today, the levels of sensitivity and the speed with which methods have to be developed and executed are increasing dramatically. So the levels are dropping down into the attogram range in some cases, and the times are dropping. The number of components are increasing that are supposed to be made with each measurement. And of course, that's bringing us into mass spectrometry for the analysis of pesticides. Veterinary drugs are very similar. The number of antibiotics is growing. The number of steroids and other growth hormones, both legal and illegal, are growing. And their measurement, identification, quantification is becoming much more dramatically in the public eye and in the laboratory eye. So again, this pushes us to mass spectrometry. Trace metals, the levels are going, not only do you have to measure more trace metals faster, but you've got to do it at low levels. And not only that, you have to do many trace metals in the presence of massive amount of other metals like sodium or potassium. So there's a lot of, the chemist is being pressured from both time and skill and the magnitude of the task in terms of both throughput and technical difficulty on every one of his analyses, and that's pushing the world into mass spectrometry. In addition to that, there's other new compound classes. Not that the compounds themselves are new, but their presence in the food safety arena is new. Mycotoxins have become, from the simple B1, G1, B2, G2, the aflatoxin series, now all of a sudden there's hundreds of mycotoxins that are being considered for monitoring and surveillance. Steroid hormones, again, the impact of illegal hormones and so illicit hormones is rather significant. Marine toxins are growing in prevalence as the beds in the producing countries are becoming more and more mature. Dioxins in food is becoming a very prevalent assay, and of course everybody is aware. Anybody who has a elementary school child in North America and Europe knows the impact of allergens and the impact of that on their daily lives, especially in school. These are all emerging applications. All of them are low-level assays in difficult matrices, and that drives the need for mass spectrometry. In addition to the chemical component, the food industry is not just chemical measurement. There's a huge biological component. Pathogens, for every outbreak of chemicals, or every chemical outbreak like melamine or ethylene glycol or anything like that, there are at least 20 to 30 outbreaks of the biological kind. And possibly even more, depending on the country. So the bacterial contamination as biological contamination, pathogen contamination is rather significant. And again, there are ways that mass spectrometry is being used to quickly and effectively monitor pathogens. So in a large way, the measurement and identification of pathogens today is largely done the way it was done with Pasteur a long time ago, over 100 years ago. And that is a significant issue in today's modern world in terms of throughput and sensitivity and the legal nature of today's world. In addition to that, commercial issues like species identification, identifying in a frozen block of white fish which fish species you're actually paying for and what you actually got. That's another thing. These are all biological type applications that mass spectrometry is being used to help develop methodologies. And then there's the issue that in every food safety environment there are the things that are going to be, that might be a problem, and then there's the things that come completely out of left field, like melamine. So the question in people's minds when they work in a food safety environment is, what is the next thing that I don't know about that's going to become the next crisis? And in those environments, we're talking about discovery of unknown chemical contaminants. So in these cases, we use TOF. Agilent Technology specializes in TOF. We've concentrated on that technique. We've tried others, but we've concentrated on TOF techniques for a variety of reasons. And we use these for our compound discovery environments, looking for unknown, unexpected compounds in food matrices. And the nice thing about TOF is it's very amenable to very, very novel data mining techniques like MPP and personal compound database and things like that. Now, those are our trade names, of course, but these are very novel database mining and data mining techniques that use the high data density of TOF and the precision and accuracy, mass accuracy of these techniques to be able to mine data in ways that we were not able to before with nominal mass instruments. And so in terms of spectrometric power, we can make a few general observations. Triple quad instruments can very selectively measure down to 200-300 compounds at a time at the sub-picogram level with either GC or LC systems. And most credible manufacturers have systems that can do this. And depending on the nature of the instruments, you can get dwell times in capillary GC on GCMS triple quads that are down to a millisecond, with settling times in the microsecond range. Polarity switching can now in LCMS can be done in tens of milliseconds or even faster. You can have a sensitivity where you can actually see single ion events, and you can actually make methods based on single ion events if your background permits. And you can now have resolutions that in quadrupole worlds that are available that can be achieved to the order of 0.1 AMU reliably. You can do that fairly effectively. Not with all manufacturers, of course, but it can be done. In terms of the TOF-based instruments, you can measure thousands of compounds with resolutions that exceed 30,000 at half height, and with either GC or LC-based systems. And with the mass accuracies and precisions are better than 1 part per million routinely, and you can scan in excess of 100 scans per second. And in some QTOF embodiments of the TOF-based technologies, you can have MSMS capabilities. And hold on a second. Oh my. Now, in addition to that, you have trap-based systems. Now, Agilent Technologies does have trap-based systems, but not to the same degree as as some of our competitors. We have trap-based systems have abilities in many embodiments similar to TOF-based systems. The scan experiments, however, are done in time rather than space. So long lists of MRM experiments, depending on the type of trap and its design, can take longer than chromatographic timescales would find appropriate, especially in gas chromatography. Maybe not so much in LC, but in gas chromatography they become limiting. But you can do much more complex experiments in trapped instruments because you've got the molecule trapped and are able to query that molecule in a variety of different ways. So you can develop much more complex applications. So we come up with, internally at Agilent, we've come up with a technique. And basically we say, when you're facing a problem, you decide whether you know what you're looking for. And by know what you're looking for, if you don't know its name, its mass, and its chemical structure, you really don't know. So I'm looking for pesticides is not knowing what you're looking for. Okay? If you're looking for dieldrin, then you know what you're looking for. In those cases where you're doing that, you know what you're looking for, you're typically interested in whether it exists and how much is there. And in those cases, a triple quad approach with MRM is basically used. In other cases, you're not sure what's there. You have a suspect list. You may suspect what's there, but you're not exactly sure. Two different approaches can be done. Either a triple quad approach where you have very large MRM databases and you go through the MRM lists and in terms of hundreds of MRMs can be done to survey the sample effectively. Or you can use a TOF approach. And you can use a technique where you're using a scanning and data mining combination of techniques with MS-MS confirmation later. And of course, you wouldn't expect it in an Agilent slide, but this similar thing can be done with traps as well. And then you have the discovery, where you have absolutely no idea. You know something is happening. You know, fish are turning to frogs. All the reindeer are turning female. Something of that nature is happening in the biological world, and you're going to try and find out what is going wrong. It could also be something pretty dramatic and not funny at all, like dogs and cats are dying and other things like that. Humans may be getting ill, and you know, you can't make light of that situation. And in those cases, you're into a discovery mode. You may not know. You can run suspect lists forever, but you may have a new compound or a completely unexpected compound, and then you have to discover what that compound is, and you're into a compound discovery mode in a food extract. So in those cases, we used time of flight, and other competitors would use, for instance, ion traps or Orbitraps or what have you. And realistically, in a discovery world, all bets are off. You use anything that you can actually use to actually to understand and give you a clue. Sometimes that could be NMR, sometimes it could be LC-NMR, sometimes combinations of POF and NMR, sometimes spectroscopy, anything that you can do. Sometimes actually injecting it into a mouse and watching how many mice die. Anything you want to find in the discovery mode is open for use. And so, again, here is a little bit of a plug, and of course it's true that only Agilent has chemical and biological technology measurement systems to support a comprehensive food safety program. I can say with more certainty that we're willing to put the time and money and talent towards finding and developing new food safety applications, and we do that in a novel way. And I think we have a very extensive collaborative environment. And I have to say, after several years of doing this, the best ideas for how to move food safety forward are not going to be found in the esteemed walls of either ourselves or our colleagues, but they're going to be found in the people that do this for a living every day. And if you have an aggressive, collaborative environment inside the company, that's the only way you're going to move technology forward, because it's the only way you're going to be exposed to novel problems as they occur. And that's the center and the basis of our marketing effort. And with that slide and that last comment and this last plug, I would like to say thank you very much.
All right, thank you, Paul. For those of you who may have missed my opening remarks, this webinar will be archived on Lab Manager's website for 6 months. The link to the archive webinar will be emailed to all of you sometime next week. The email addresses of our speakers will be shown to you on our the final closing slide. And if you need a copy of their slides, you will have to contact them directly. So please take the time to note down their email addresses. Let's now turn it over to our next and final speaker, Dr. Jonathan Beck, who is one of 2 marketing managers responsible for Thermo Scientific's environmental and food safety LC-MS marketing efforts worldwide. He has been with Thermo since 2005 and has been working in the environmental and food safety markets since 2007. Welcome, Jonathan.
Okay, can you see my slides okay?
Yes.
Okay. All right, let's see if I can click through these. So thanks for the introduction. I am one of the 2 marketing managers for environmental and food safety at Thermo worldwide, and I'd like to show you some of our solutions in the mass spec area for food safety. All right, I'm having the same problem Paul did, I think, on the slides. There we go. Okay. So first I'm going to focus a little bit on the food safety arena and how we see that similar to what Paul did in his presentation, and then I'll talk a little bit about a couple of our solutions, quadrupoles, and our Orbitrap technology. So around the world, we're seeing a lot of new food safety issues, as Paul mentioned earlier. The latest one I recall in the United States was the situation in our orange juice. Oranges were being imported from South America, and they contained a banned pesticide that was detected. It was banned in the United States, and because of some winter problems with the oranges, we were importing more oranges than we normally did. and there was a little bit of an outbreak of this pesticide. So that kind of caused a scare. And this gets out in the public knowledge very quickly these days in our 24-hour news cycle. Here's a couple of examples where that has occurred. When these do occur, a lot of people know about them. It's not just food producers and grocery store managers and things like that. We all hear about them, so they're definitely things that get into the public knowledge, and we want to be able to ensure safe food around the world. Now, as our world is becoming smaller, we are importing and exporting foods from one part of the globe to another, which leads us to a lot of different types of foods that we need to be testing, as well as a lot of contaminants that we need to be monitoring. Traditionally, we would look for pesticides and herbicides in our fruits and vegetables, but that's now been expanding to things to include such compounds as mycotoxins, plant toxins. These are naturally occurring toxins that occur on the plant themselves or because of some organism on the plant. They are not added by man. There's also the concern of packaging contaminants, things like BPA, which recently was in the news in baby bottles and Nalgene bottles, and water bottles, things like that. We've got the naturally occurring marine toxins that can occur from algae blooms, red tides, things like that. If those get into our oysters or clams, they can cause severe sickness in humans. There's also disinfection agents. During the food production process, there's cleaning that needs to go on in the tanks. For example, in milk, dairy farmers need to ensure that their holding tanks are free of any pathogens, so that they're treating that with a disinfection agent. We don't want that to go into the food supply, of course. So at Thermo, we have triple quad technology, just like Agilent does and many of the other manufacturers. We have 3 different triple quadrupoles that we offer right now. The difference between them is going from the left to the right. The TSQ Quantum Access Max is the model we call the instrument on the left. That's the least sensitive of the 3. However, it is generally sensitive enough for most of the food analysis, depending on your sample preparation techniques. As you go to the right-hand side of the slide, we increase in our sensitivity of these instruments. And, of course, the price does go up with that improvement in sensitivity. Between the 3 instruments, they're all used in the food safety industry. We have customers that are using the Access Max as well as the Vantage. So it oftentimes depends on what your particular application is as to the instrument that is chosen for your analysis. Some of the common analysis that are performed on our triple quadrupole instruments around the world for the food industry are shown on this slide. So chloramphenicol, which is an antibiotic that is banned for human consumption in the United States and much of Europe. So we look for that in pork, honey, and milk coming from countries where that compound is sometimes used illegally in the food production. Same thing with nitrofurans. Malachite green is a disinfectant or anti-algae compound that is sometimes used in fish tanks, and that can cause trouble if it gets into the seafood that we ingest. Honey, tetracyclines that are used to treat the honeybees, and of course pesticides in fruits and vegetables. And as Paul mentioned earlier, the melamine in milk. I have heard some reports that there is possibly a new compound that is being put into milk and dairy products to change the nitrogen content. It's not melamine, but it's something else, so there may be something coming down the road in terms of that product as well. So some of the challenges that we face when we're analyzing a wide variety of compounds or a multi-residue approach to food safety is that we have a lot of different chemicals in a mixture, and they may not all have the same chemical properties. So everything from nonpolar molecules to highly halogenated compounds such as DDT, which is shown on this slide, still used in certain parts of the world. So you've got to consider, do these all work together well in the same analysis on the same instrument? You may have to choose different instrumental approaches, different sample preparation techniques to analyze these compounds. There's not one perfect instrument that will analyze everything that you need to be looking for. So we still have that challenge. Around the world, we have different regulations that need to be monitored. In China, there are different detection limits and limits of chemicals that can be permitted for export or import, and the EU has different levels, and again the United States, Japan, every country might have different levels. So we need to be cognizant of those different levels around the world for different classes of compounds, as well as what matrix they may be in. For the top compound, serofloxacin, the levels in fat are twice as high as the levels in muscle tissue in China, for example. Like I said, the detector choice can change based on the detection limits we need to reach, as well as the compounds we may be analyzing. And this chart kind of gives just a general overview of sensitivity versus the MRL detection limit. Going from the lower left over to the upper right, if we're going from a screening type quick approach, is it there or not, really quickly, we can use oftentimes something like ELISA, which is an antibiotic type approach to look for these compounds at higher levels. If we need to really drill down and get to lower sensitivities, we oftentimes have to go to GCMS or LCMS and MSMS instead of single quads to get to that detection limit. And then we have all the food in the world, which is a huge, huge problem when we have to clean up the sample before we put it in the mass spectrometer. Unfortunately, we just can't take a grape and stick it in a detector yet and then analyze that. We somehow have to crush up that grape and get the sample into a form that we can put into the instrument. So there's that challenge as well. We have the consideration if our compounds bind to the matrix. In other words, does it stick to the matrix and not get into solution when we're doing the extraction? So you may have to have a special extraction procedure to ensure that that's not happening. If it stays on the matrix and you don't get it into the mass spectrometer or other detector, you would think that the compound is not present in the sample when actually it is and it's sticking onto the matrix. And then you have, of course, the different LODs and LOQs, limits of detection, limits of quantitation around the world. Here's an example I just wanted to show of a typical sample preparation technique that is done in a food laboratory. This is for the analysis of 45 different pesticides in fruits and vegetables, and the limit of quantitation was at 10 parts per billion for this food product. So the sample preparation techniques are shown on the right-hand side of the slide. They would take 10 grams of the sample, sonicate it, filter it, add some chemicals along the way, evaporate it, add some methanol, reconstitute the evaporated sample, and then finally dilute it, filter it through a membrane, and then inject it onto the triple quadrupole instrument. So you can see this is an extensive sample preparation scheme that is required for the analysis of these compounds in fruits and vegetables. So switching gears from our triple quadrupoles to our higher resolution instrument portfolio, we call this the Orbitrap technology. It's the similar idea, I should say, similar to what Paul was discussing with time of flights, in that we're doing a high-resolution scanning technique, but it has some differences. The Orbitrap is a trap-based technology, and I'll show you the details of that on the next slide. We have 3 different Orbitrap instruments that we offer right now. On the left is the Exactive LCMS. In the middle, a Q-Exactive LCMS-MS, and then the ultimate hybrid system, which is an Orbitrap, which is an ion trap in front of the Orbitrap. So that's a hybrid instrument that can be used for experiments such as the discovery type experiments that Paul was talking about in his presentation. In the food safety arena, most of our customers are either using the Exactive or the Q-Exactive, and I'd just like to spend a minute and tell you what the main difference is between the two. You can probably guess from the difference in the name, Q-Exactive versus Exactive. Q stands for quadrupole, and that is the main difference between the two instruments. On the left-hand side is the Q-Exactive, and you see the quadrupole mass filter that is inserted in between the C-trap and the ion source. So what we're able to do with the Q-Exactive is to pre-select ions before they enter the Orbitrap mass analyzer. So that allows you to filter out matrix or other compounds and just get certain masses into the mass spectrometer, the Orbitrap mass spectrometer. With the Exactive, everything that goes through the source enters the Orbitrap mass analyzer. So you get an additional degree of segregation with the quadrupole mass filter in front of the instrument. So this is analogous to a Q-trap if you've heard of that kind of instrument, compared to just a Q-TOF instead of just a TOF. We add the quadrupole in front. So the difference between those two instruments, the Q Exactive does have a higher resolution. It will go to a resolution of 140,000, where the Exactive tops out at 100,000. Typically, our customers aren't running at that high of a resolution. Routinely, they're running more at about 50,000 to 70,000. The scan speeds you can see here, this Q Exactive does scan faster than the Exactive. And there are a couple additional scan functions that are available on the Q Exactive instrument. That's because there is a quadrupole in front of the Orbitrap mass analyzer, so you can do some additional experiments. So how does the Orbitrap Exactive work with a food sample? This is a sample that we used from the California Department of Food and Agriculture. They provided us with a matrix of bell peppers, and we spiked some pesticides into there. We did a simple cleanup, the catcher's technique, and we had our calibrators and our standards added in the range of 500 parts per billion down to 1 part per trillion. And we did a targeted data-dependent MS-MS. So what we did was we knew the 62 pesticides we were looking for. When we saw one of them eluding from the column, we told the mass spectrometer to use the quadrupole, isolate that particular pesticide, and then analyze its product ions in the Orbitrap mass spectrometer. We ran this at a resolution of 70,000, and we in the confirming MS-MS scan, we ran at a resolution of 17,500. And I won't tell you all the HPLC conditions, because that's not really important here. What we wanted to show was the difference between two calibration curves. There are actually two curves shown on this slide. We ran one in pure solvent and then one in the bell pepper matrix. And we laid them on top of one another. And what we were looking to see was, if we're in a matrix, sometimes you'll get matrix suppression or matrix enhancement. And what this is showing is they lay over one another perfectly, meaning we didn't see that effect. So we had good cleanup in our catcher's extraction, and we're able to quantitate quite reliably down to the low levels. On the left, you see a chromatogram for one of the compounds, azoxystrobin, at a level of 1.5 parts per billion. One other thing we can do is speed up our analysis. This is an example of UHPLC. You may have heard of that from various manufacturers. What we're doing is running HPLC conditions at a very high flow rate, which then generates a high back pressure. But what we're able to do is utilize small particle sizes and separate our components in a short amount of time. The original analysis for this took 18 minutes. We've shortened this down to a 5-minute runtime, and all of the compounds are eluted in less than 1.5 minutes. And the nice thing is that the Orbitrap instrument, the Q Exactive in this case, is able to scan quick enough to monitor all 62 of those pesticides in less than 1.5 minutes. And that'll be my last slide. Just want to point out we do have a food safety website. It is shown at the bottom of this slide, www.thermofisher.com/food safety. That's all one word. You can go there and see a lot of application notes and presentations on our solutions for food safety. So I encourage you to go there, and if you can't find that website or you don't have time to write it down, feel free to email me and I can direct you to that.
Great.
And I think I hand it back over to you, right?
Yep, absolutely. And as you can see, the email addresses of our speakers are now on your screen, so make a note of that. And if you need a copy of their presentations, or if you need to ask them, more questions, there it is. But we do have some questions that we have received from the audience, so let me get to that. But before I go to the audience questions, let me ask you both, Paul and Jonathan. So how exactly do you work with customers when they email you or when they contact you? What are the types of services that you provide?
Should I go first? This is Jonathan from Thermo. Yeah. Okay. So we have a food safety team, if you want to think of it that way. So we're able to offer everything from assistance on method development. If you were to buy a new instrument, we have a team of specialists that can come and help you out in your lab. They're experts in everything from our sample preparation team to our HPLC team or GCMS. If you need to optimize your chromatography techniques, we have people that can come and help you with that. when you purchase one of your instruments. We also have something called the Food Safety Resource Center, and that is based in Germany. And what their responsibility is for focusing on these emerging issues. For example, that orange juice scare that I talked about earlier. What they did is they will monitor the news, and when there's a new technique, or excuse me, a new contaminant, it's their job to quickly come up with an analysis for that if we don't already have one. So they worked very quickly and had an analysis ready in just a few days. So we have that. So we do have a lot of different support in that way, methods that we can supply to the customer as well. A lot of our analysis has already been done by our demo labs or something like that, and it's no need for you to reinvent the wheel. If we have that, we can help you get started. So we have quite a few different resources at Thermo.
All right, that's good to know. Yep.
Yeah, well, for Agilent Technologies, in food safety specifically, I run a group of about 25 scientists worldwide. We have laboratories in application laboratories with LCMS, GCMS, and either ICP-MS or biological analysis in India, in Bangalore, in Beijing, in San Francisco, at UC Davis, and in Santa Clara at our site there, in Delaware, in London, and in Waldbronn, Germany. So around the world, we have fully staffed laboratories that are able to address any analyses that are brought to our attention in the food space. In addition to that, this is just what we call field research. Basically, on the lookout all the time with some of the world's finest food safety researchers, and we're looking for the next new thing. The next problem, or how do we do today's problem better? So it's basically field research. In addition to that, we have field resources and field laboratories in virtually every major economy, and that's they have their dedicated individuals that are able to give support with food applications in both gas phase, liquid phase, and ICP-MS and biological, as a matter of fact. And then, of course, you have the sales force with its resources and both the sales and repair individuals. So there's my small group alone of 25 people has a large number of laboratories dedicated directly to answering food safety questions. And so we work with customers in a variety of different ways at a variety of different levels in the food safety chain, from before the problem hits to during when the problem is hitting to after in terms of surveillance.
Right. No, can we spend some time just talking about sample prep, because I guess this is huge as far as food safety is concerned. So how do you work? Do you guide people with regards to sample prep as well? What are some of the key issues that you have found associated with sample prep or sample carryover or cleanup, and how can people minimize some of those problems?
Okay, let me, Jonathan, do you mind if I go first now?
Sure, we'll just switch back and forth, it's fine.
Alrighty, yeah, it's fine. So in terms of sample prep, we have a sample prep division again, within most major geographies. We have laboratories that are dedicated not to the instrumental part of the analysis, but specifically towards measuring, coming up with new techniques. For instance, we brought out a catcher's line. We have multiple chemistries based on catchers, and these are done in conjunction with our research scientists in the field. Usually today, catchers in produce is largely the approach people use, especially in conjunction with triple quad mass spectrometry. But there's a huge line of solid phase extraction products, both dispersive and in column format. There's prep LC approaches to sample prep with fraction collection. There are, again, a wide variety of sample prep. And once you have the sample prep technology, then the question becomes one of robotics. We have a robotic division that effectively can take a sample prep chemistry and move it through from, beyond the ability of an individual to do 10 or 20 samples at a time and move it up to 200 or 2,000 at a time.
Okay. Jonathan?
Yeah, sample prep is always my least favorite part of the experiment. I never like doing it, and I would argue that it is probably the biggest challenge in food safety analysis. You've got everything from very complicated meat-type samples or very, very dirty matrices to things that are a little cleaner, but you still have to clean them up. And as Paul said, catchers is kind of the technique of choice these days, but sometimes that's not applicable for everything. If you have high fat containing samples, for example, it doesn't always work the best. So there are different ways of doing it, and we do have quite a large consumables and sample prep division as well that has many of the same offerings. And another thing that we offer is something called turbo flow technology. What turbo flow technology is a online sample prep technique. So if you had a food sample that you ground up in a blender, for example, and did a small dilution on that, what we can do is take some of that sample preparation technique that you would typically do with solid phase extraction offline and bring that online. So it's part of the auto sampler technique. We do the extraction just before we introduce it to the HPLC column, and we do that online. The advantage of that is you don't manually have to do that offline sample preparation, and the reproducibility is very good because you're letting a robot take over in the auto sampler and doing that technique over and over again. So that's some of the areas where we've seen a lot of people have a lot of interest. It's the online sample preparation technique. And it definitely saves a lot of work in the laboratory. You have a little bit more expense up front with some additional hardware, but it pays for itself in a short amount of time when you're running a lot of food samples.
Okay, thank you both. Well, here's a question. It's a little elaborate, but bear with me as I read it out. We have one of our attendees who has heard of mass spec used to identify selected intermediary metabolites for pathogenic bacteria. This approach would both detect the bacteria and identify the pathogen. This should be very fast, very selective, and very sensitive. So why haven't I heard more about this technique?
That's a really good question. Jonathan, you want to go first?
Oh, I was going to hope you wanted to go first on it.
Oh, all right, no problem. I'll take that. Okay.
It's in our chat box too, Paul, if you need to review any of it.
Yeah, no, that's okay. It's excellent. That's an excellent question. Okay, and for those people interested in pathogen, in the instrumentalization, if you will, of pathogen ID, you can remember back in the early 80s, Hewlett-Packard brought out MIDI, which was based on a technique using identifying bacteria through fatty acid profile in the cell membrane. A lot of the techniques that are involved with the identification of bacteria using either proteins or any of the metabolic characteristics largely depend, and we've had those techniques. We've had that back in the early 80s, and we've had subsequently prototype research efforts to see if we can do that. But generally what happens is that unless you standardize the growing conditions of how you grow the bacteria and the nutrients and all the rest of it, and you can standardize that across a large number of laboratories, across a large number of geographies, you end up having too much variability to be able to get you a identification at the serovar, the genotype level. Yeah, it is true that those markers are very selective, and there are a variety of techniques from GC and DCMS and MALDI and even FTIR on a culture dish. It is possible to do so, but the problem is, depending on how they're grown, small variations can creep up into the measurement, and it could effectively negate the power of the technique. So while we are always hoping that some simple technique could actually come out and be able to do that by looking at some marker, the reality of the matter is that the DNA is the marker of interest in pathogen ID, and we've come to that realization after, how many years is it now, since 1983? Many years of research in that area.
Thank you.
Yeah, I was thinking that this is probably the approach that's taken out at some of our big governmental labs where they do have these very controlled environments. And what we see in a lot of our food customers, and I'm sure it's the same as you, it's smaller labs. It's labs that need a result very quickly. Yeah. Oftentimes they're not in a laboratory that you'd see the controls where you can do this reproducibility. It's oftentimes down at a shipping container port or something like that where they need these results turned around very quickly. Yeah. So yeah, it's kind of still in the research realm, I would say.
Yeah, the people that need it the most are the least likely to be able to have the same conditions as the large research institutions that can do the thing, where they only need the data in-house. So they only need self-contained data, and within their own infrastructure, they can identify bacteria pretty effectively. Once you open it up, then all of a sudden it falls apart. It's unfortunate. We were hoping we had the world by the tail in the early '80s. It didn't turn out to be that way.
Interesting. Okay, well, here's another interesting question. We have someone who has developed a Maldi-Toftoff approach for detection of bacterial toxins produced by B. cereus, the bacterium. I have used FTMS to select toxin markers in order to synthesize stable isotopes of toxin peptides for the enterotoxin detection. And I was wondering which LCMS method is best for quantification and how an MRM approach will increase the value of our quantitation approach.
Well, that's a good one. You go ahead, Jonathan. That's an excellent question.
Okay, so most of the time when people want to go to an MRM approach, they're going after there's a couple of different reasons why. The first one is that it gives you more selectivity. So you're able to just hone in on what you're looking for. So that's the first reason people will go to it over a full scan approach on a triple quad, for example. If we're doing a full scan single quad, and then we go to an MRM approach, we're able to increase our order of magnitude, at least in our sensitivity, because what we're doing is lowering our noise that's coming into the instrument. Now here you're doing a peptide analysis, I think, if I'm reading it correctly here, and you're asking which LCMS method is best for quantification. So if we're doing quantitative work, that typically falls in the triple quad arena. However, we can get very excellent quantitative results on the Orbitrap type instruments as well. So that's not really a problem. I guess another consideration is your mass range. If you are running a triple quad, typically your upper mass to charge ratio is somewhere around 3,000. Depending on the manufacturer, that could be 1,500 or something like that. As long as you have a multiply charged ion, if it's a higher mass than that, you can still analyze that. But yes, an MRM approach would give you that increased specificity. If you're going to an instrument such as a TOF or a Q-Exactive, you're going to get that high resolution capability as well that will help with your quantitation, making sure that you're getting the quantitative result for exactly what you're looking for because of the high resolution technique. And yes, we offer that kind of analysis as well. I'm sure Agilent does. I'll let Paul answer that.
Yeah, that's an excellent question. We've done projects like this on a few toxins. The first one being botulinum toxin, from Clostridium botulinum, and one of the things that you use generally to start off with a general proteomics discovery approach to actually discover it. And once you do, then typically you go into a quantitative proteomics approach. Now you can use, of course, you can use an Orbitrap or a Q-Exactive, or you can use a Q-TOF. The idea is, or a triple quad. Now, a triple quad would mean that you've gotten your characterization of your peptide or your protein and the digestion products that you would use in a peptide analysis. You've characterized that to such a degree that you know exactly what you're looking for. You know all the peptides, not just some of them, because you don't want to make a mistake. You want to know all the major peptides and quantitate all of them, and then do a quantitative measurement as a sum. That usually helps in quantitative proteomics approaches. Those approaches are very well known, and you can also use them if you then choose to make a peptide, to make a protein, synthesize it from isotopically labeled peptides. Then you can also tell the differences, and that carries into the execution of a measurement for isotopically labeled peptides as well. So generally, both the Orbitrap and the QTOF would work in the early discovery time frames, and then you can transition into a peptide quantitation approach in electrospray. That'll bring that brings mass to charge ratio down where you can actually use it with a triple quad. The other approach is MALDI, or you can actually go into a vacuum MALDI approach. And I have to say that while I have customers that have a lot of expertise with that, my expertise runs fairly low at vacuum MALDI. So I can't put that technique into perspective other than in the early discovery stages. Quantitation by vacuum MALDI, I don't think you can approach the quantitative proteomics capabilities of modern triple quads. But I could be wrong.
Well, that's a separate discussion, I guess, comparing those two. But here's another question, slightly more generic. Could both of you just comment on some of the common background influences that come into play, and what is a general solution to some of these problems with the LCMS analysis?
Who went first last time, Jonathan?
I think I did. It's your turn, Paul.
Oh man, thank you so much. Yeah, but with background is a very great concern when you're doing discovery work, when you're actually working in MS mode. When you're working in MS mode and you're trying to discover, look for something you don't know, or use a general MS scan approach, whether it involves a trap or whether it involves a TOF. Anytime you're working in scan mode, the background is really significant. In MSMS mode, background, effectively chemical background, gets driven away by the selectivity of the MSMS approach, but still significant. You still can't throw it away. If you have a background like humic acid that has an ion at every mass, and it turns out to have probably a product ion at a mass that's extremely inconvenient for the thing you're interested in. But typically, if you're interested in what kind of things can impact background, of course, mobile phase. Look, if the water's bad, if you've got white fungus growing in your water bottle, your background's going to be high, whether you're doing it in MS or MSMS. And I've seen that in the jungles of South America, believe me. And your solvents, of course, have to be the highest purity and all the rest of it. Now, assuming that you do that, then you have the chemical background in the matrix itself. And of course, that's all the general, that goes to the general cleanup problem. When you're dealing with fruits and vegetables and most produce, you've got tons and tons of glycosylated flavones, isoflavones, anthocyanins, color bodies. You've got tons and tons of carotenoids, all kinds of chlorophylls. So you have a tremendous amount of chemical background. And the objective then is to pull chemically the majority of that background away from the target compound, and then use chromatography in some way to help fractionate in a manageable way, help fractionate a lot of the components of that background into a manageable methodology. And it's really difficult to say what the specific backgrounds are being in a matrix. I mean, it's all matrix dependent, and we'd have to have more detail about what matrix you're looking at.
Jonathan, I know you touched on matrix issues.
Yeah.
But anything else?
Yeah. Yeah, so in addition to what Paul said, that example I gave with the bell peppers is kind of getting to that point. If you're back, you may not be able to separate it. In most cases, you can't get rid of everything, no matter how good your sample prep techniques are. And it can contribute to things like the matrix enhancement or matrix suppression. In that case, we didn't observe a lot of it, but that's certainly always a concern. One of the other approaches that is now taken, and we bring in a little software into the game, if we're doing a full scan experiment on one of the high-res instruments, whether it be an Orbitrap, Q-Exactive, Exactive, or a TOF instrument, Some of the games you can play, and we all have software that does this, is if you have something, a compound or a reference, if we want to call it that, a reference of your grapes that you have, and then a sample that is suspect, and you want to see what the difference between those two is. Chances are the things like chlorophyll and all the things that are in there in a regular grape sample are going to be the same from one grape variety to the other. So if you have a reference and you have a suspect sample, what you do is compare those two samples. And we all have software that can handle this, and we compare those two chromatographic runs, and what we do is look for the differences. And a lot of times in the differences, that's where we find our problems or our contaminants or what has changed. So a lot of powerful software can utilize the high resolution plus the all-ion capture of these scanning instruments. You can't do this on a triple quadrupole. This is purely in the realm of the high-resolution instruments, and you have to be in the full scan mode. But that's one way to kind of do a background subtraction, because you're never going to get rid of it. But the things that are there, hopefully they're the same from one grape to the other. You're interested in what changed here from this sample to the next.
Yeah, and that really is sophisticated background subtraction indeed. I mean, it's well beyond what we considered, what's generally considered background subtraction, whether it be the software on the Orbitrap or the QTOF. And the thing that is implicit in what Jonathan's saying is that the software, the old software idiom of garbage in, garbage out stands there too. If you have bad spectrometry or you have insufficient chromatography or high suppression or what have you, you're not going to correct for that with data mining techniques. You've got to do your homework up front. The sample's got to be, you got to give the compounds that you're looking for a chance to show up against background spectrometrically. And once you've done that and they're well resolved, and hence we give the high resolution, or whether they be resolved chromatographically or whether they be resolved spectrally, in either case, you've got to give the peak a chance to poke its head up above background in a way that can be recognized. If you've got a big smear and everything runs into each other, regardless of resolution or chromatography, well, you got to go back to the lab bench and do some cleanup, because you need another degree of specificity there to give the software and the technique a chance to be able to find your compound.
Yeah, and chromatography is not going anywhere. It's not disappearing. I know a lot of people are interested in techniques that maybe no chromatography, or they just inject a plug of sample. But as Paul was saying, you got to give that peak a chance to come out. And if it's just going in with everything, the most powerful software in the world and highest resolution instrument is still going to have a lot of trouble with that.
Yeah, Jonathan, I've got my first triple quad in 1991, and 1991-92. And I remember back then everybody was saying, oh, these instruments are so impressive, you don't need any chromatography. And the world's been trying to prove that ever since, and every year there's still a chromatograph sitting in the front of it, trying to simplify the job. It's not going, as Jonathan said, chromatography is not going away anytime soon, especially in the food business. There may be applications where it might, especially maybe in pharmaceuticals or what have you, but in the food business with these complex samples, you need every measure of discrimination you can get, and a chromatograph is a very inexpensive way to put fractionation power into your problem.
Yep, very, very useful advice. Well, let's tackle this one. CDC has come up with a MS method to assay for the presence of the Shiga toxin by monitoring the depurination of the large ribosomal RNA. Yet I haven't seen this method in such official methods repositories such as the AOAC. So have any of you worked out methods for this?
Yeah. Jonathan, I think you're up.
Okay, I'm not keeping track.
You sound like you had something to say. Go ahead, Paul.
No.
Or do you want me to go?
No, look, this is that approach. That is an exceptionally advanced approach. For that to make it into the routine hands of people in validated multiple, multi-ring trial type environments is going to take some time. That's an advanced technique.
Yeah.
That laboratory, that laboratory at the CDC, I don't know that the CDC does. If everybody can do it, they wouldn't touch it. So by definition, if something comes out of that laboratory at the CDC, is by definition one of the most advanced technologies you can have. That doesn't necessarily translate into a ready-to-use methodology in the way that pesticide analysis or mycotoxins can be done. I mean, those are really highly advanced techniques, and it's going to take a while.
Yeah, I would say that the agencies such as AOAC or FDA or the comparable agencies outside the U.S., they try to err on the side of making a method that is accessible and reproducible by a wide variety of labs. And something like this out of the CDC, as Paul said, is a very technical approach, and they don't want to make it where you have to have $1 million worth of equipment to do the analysis in most cases.
Now, Jonathan, you and I would both appreciate if that was true.
Yes, that would be fine.
Because that's how we pay our salaries. But as Jonathan knows, we spend our lives trying to take technology, the cutting edge technology, and pushing it out to the practical user. And some of these technologies are really highly advanced. So especially when you're talking about a ring trial like the AOAC, the first thing the AOAC is going to do is going to put a request out. How many people want to participate in this trial? And then they're going to say, well, what kind of equipment is involved? And now you're back to one laboratory or two laboratories in the world that can compete. Now you've got yourself a problem. So it's a difficult thing. And you find that a lot of laboratories have some very innovative approaches that work, again, just like the previous question, that work within an isolated environment, but have difficulty being pushed out through, and they will get there, but it will take some time.
Okay. Well, actually, for a moment there, I felt like I was moderating Car Talk, that popular radio show, without the annoying laughter, of course. But yeah, this is good. I'm sure it's really good advice for people out there. It's really it's just very informative. But as a last question, and this is from me, I want to ask both of you, what areas do you think users can expect to see more changes and more improvisations in the near future?
Yeah, I think we. Do you want me to go first, Paul?
Yeah, you go ahead.
So what's on the horizon? There's always new I want to say source technologies. For example, we have been working with a company called IonSense, and it's what they do is offer a way to analyze samples in real time. What I mean by that is you're able to actually take a commodity, whether it's a banana or an orange or something like that, or swab a food sample, and then introduce that to the mass spectrometer with different or little or no sample prep. Things like that can be used in a screening environment. We're seeing some success with that, and I know IonSense is a company that sells these technologies to all the mass spec vendors. So what I see is this approach to, we need to get quick results. We want a result pretty quick, and we want it in a challenging environment. And when I say that, I mean the guy down at the Oakland port who's getting a shipment of bananas from Brazil, he needs to say, the FDA inspector, is this okay or not? And we can't let those bananas sit around for 2 weeks as we send the samples over to the lab in Florida and they send it back. So these technologies that allow for this rapid screening is an area that's taking off a lot. I think that that's kind of one thing that we're seeing a lot more, bringing the mass spec to areas where it hasn't been before, getting it out of the big labs and getting it down to the more routine testing areas. That's,
interesting.
Where we see future, not just necessarily with mass spec, but in food safety and the analyses of food safety issues by mass spec, it's along a few fronts. Certainly out of lab is definitely one of them, and we all introduce products of out of lab, new sources, etc. So you have the instrumental power access where you're going to get increased sensitivity, increased resolution over time, a more selective ionization, those kinds of approaches on instrumental power. On another axis, you're going to get the impact of sample prep. Sample prep is the great, how should I put it, the great frontier. And sample prep, and we're making a lot of, as everybody is in the business, making a lot of investments in sample prep. So I think you're going to see more selectivity in sample prep. You're going to see much more effective, cheaper, faster techniques in sample prep that are more selective. Maybe more available chemistry, certainly, but you're going to see more sample prep capabilities. You're going to see more automation of sample prep, certainly. And the next thing you're going to see centers around the impact of DNA, the pathogens, the measurement of and identification of pathogens based on the instrumentalization of microbiology. And mass spec is going to play a very large part in that. So I think that's what I think the future will hold.
Okay.
I have a friend who lives in China. He's an American friend, and he keeps challenging me to invent the handheld mass spectrometer that he can take with him when he goes out to eat dinner, so he can test that his food is safe. So that's the holy grail.
Yeah. We're all trying to invent the tricorder, aren't we?
Yeah, a handheld instrument that will deliver results directly to your mobile device.
Yep. The iPhone attachment.
Yeah, and if you can do it from 30,000 feet in an airplane, even better.
Yeah. Why not? Let's all be optimistic. Well, one question just popped in, and I'll make that the absolute last question for today. The FDA FSMA wants food manufacturers to validate their complex ingredients, such as green tea, for example. Yet no one wants to load this onto a column because it might destroy the column. So what about MS scans? Will such be, will they be reproducible enough to make this happen?
Jonathan, you want to go first? That's a good question. You want to go first?
Yeah. I guess that's certainly I guess one of the advantages of something like the DART technology from IonSense. You can take tea leaves and analyze them directly without putting it on a column, so you're not going to clog something up. Now that only works if your contaminants or what you're looking for are able to be ionized by putting that in a hot gas stream. In other words, it can't be contained within the sample. It doesn't. Something like that, a surface type analysis. Otherwise, you're still going to have to do some sort of sample cleanup, whether it be a catcher's type cleanup or online TurboFlow type cleanup. It's just, you're right, you can't crush up some tea leaves and then inject them on a column. You're right, it's only going to last 1 or 2 injections if you're lucky.
Yeah. What the FDA is asking on those botanicals is that you be able to characterize the material. If you can characterize the material by shaking it up in water and taking a drop of that water and putting it into a UV and taking a spectrum, they're not asking you to do very, very major things. The question is, unfortunately, that characterizing botanicals is a very difficult thing. It can't be done with a simple UV spectrometer. So now you are into mass spectrometry and you're into LCMS, possibly GCMS. Dr. Khan at the University of Mississippi has a botanical center. He has several thousand, I think it's 20,000 authenticated botanical samples. And his whole lab is dedicated towards the characterization of these reference materials. Characterizing a botanical sample that is absolutely brand new is not a trivial matter. Now, how you characterize it and all the rest of it, I'll leave that between you and your FDA inspector. But I think that it certainly can be done. You can certainly begin to do a tremendous amount of characterization, but you have to know what you're characterizing and pick a compound class that you're going to be able to home in on, and that compound class will tell the difference between your botanical and a closely related botanical. And if you can do that and you can prove that that actually does work, then you have the basis of it. But generally, without knowing the specifics, it's hard to say whether that variability or that characterization vector will be in LCMS or GCMS or FTIR or even NMR. It's really hard to know. It's unfortunate that the burden is on the characterization. You've got to characterize. it's not an easy matter to actually a priori be able to tell you what technique would actually serve that purpose.
Okay. All right. Well, that brings us to the end of the webinar. I'm sure other questions will crop up as you get back to your analysis and to your samples. And if you need more information or more guidance, Paul and Jonathan are definitely the people to reach out to. They have a lot to say, and I'm sure they'll be glad to help. Again, their emails appear on your screen, and feel free to contact them directly. On behalf of Lab Manager, I would like to thank both Agilent Technologies and Thermo Scientific for their participation and support. My sincere thanks to both our speakers for putting in the time and effort to make their presentations very interesting, very informative, and again for being so candid with their feedback and providing such elaborate answers for what are some very, very difficult issues. The webinar will be archived on Lab Manager's website for 6 months, so do visit us online to review this webinar. We also have past webinars on the website, and if you need information about our upcoming webinar topics, you will find that there as well. I hope you can all join us again in a couple of weeks when we will be discussing innovations in biological safety cabinets. I hope that that discussion is as interesting as the one that we had today. So thank you all once again for your time and attention. Until next time, goodbye and good luck.